Hello students, welcome to EPG Pathsala. I am Dr. Harin Ram Seri from Keralimal College, University of Delhi. Today I am going to talk on module Prey, Predator and Plant Herbivore Interaction part first under paper Principle of Ecology. Learning outcomes. After studying this module, you shall be able to understand describes the module of predation, understand the prey-predator system, illustrate the functional response and different type of functional response, define aggregative and numerical responses, understand the foraging theory. Introduction. Different species interact with each other to compete for food or space through predation. Predation is called as a biophagy and defined as one species feeding on other living organisms. Moreover, it's a broad term used for those species which not only eat other species but often involves in killing of other species. On Earth, as ecosystem, humans are the major predators. It is an association where mostly stronger attracts the weaker organism. However, considered more broadly, predation can be distinguished into five specific types, namely herbivory, parasitoidism, carnivory, cannibalism, and parasitism. Herbivory is a type of predation where all type of grazing animals prey on plants. This type of prey predation association does not involve direct killing, but plants, seeds or fruits eaten by the animals causes damage to the green plants. Carnivory is a typical kind of predation where predator or stronger animal prey on other carnivorous organisms or on herbivores. Another specific type of predation includes parasitoidism, where an organism called parasitoids, generally these are insects, parasitoids, predates on other insects called host insect by laying eggs near or on the host insects. These eggs hatches into larvae that feeds on tissues of host insect and subsequently killed and eaten by the insect parasitoid. Parasitism is a type of predation where parasites, plants and animals are the organisms that do not kill and consume the prey or hosts but depends on them for nutrients. A special form of predation is called cannibalism, in which predator and prey belongs to the same type of species. Certain groups of tribal people show cannibalism where on death of any family member they eat their body parts. The predator does not interact with prey only but with other predator species through competition. The interaction-mediated competition between predator species is indirect. When the prey of different or same species competes for food and space among themselves, creates indirect competition between predators. Whereas when predator compete for same prey species that is in short supply in causes indirect competition between predators. We can see in figure number first. This figure represents the direct and indirect competition in a predator-prey system. When the prey of different or same species competes for food and space among themselves, creates indirect competition between predators. Whereas when predators compete for same prey species that is in short supply, it causes indirect competition between predators. Predation is considered as a useful interaction when animal predates on pest and prey, but predation is considered as undesirable when prey is a valuable resource. Predation is an important interaction that reduces the abundance of prey and restricts its distribution. Thus, predation secondary to competition is the main type of interaction that influences the community shape, structure, and organization. Models of predation. Organisms use two types of system called discrete generation and continuous generation systems. By following two simple type of prey predator models, A. Module for organisms with discrete generation system. Discrete generation system is used to explore prey-predator interaction models. Several insect hosts act as a prey and insect parasitoids acting as a predator under seasonal environmental conditions have a one generation per year and are described by following type of models. Logistic equation describes the increase in the prey population in the absence of predators and given as where anti population size T generation time B slope of reproductive curve and anti minus n EQ difference in the population size from equilibrium size of population in absence of predator. We must subtract a term related to the number of prey eaten by predators from the logistic equation of prey under the absence of predator. The logistic equation of prey in the presence of predator is represented as where Pt in generation time t the size of predator population. C. Constant determining the predator efficiency. Assuming that the predator's reproduction rate depends on the population size of prey, the equation for predator population is given as Pt predation population size and prey population size T generation time and Q constant determines the prey utilization efficiency of predation for its reproduction. When these equations are put together and interpreted after calculating them over many generations resulted into a cycle of prey and predator numbers, we can see in figure number 2, there are formed possible outcomes of these calculated equations: a stable equilibrium without oscillation, divergent oscillations causing extinction of either prey or predator, convergent oscillations causing extinction of either prey or predator, stable oscillation. This figure represents a hypothetical classical oscillations in prey predation system with discrete generation depicting population changes. X-axis depicts the population size, y-axis depicts the time period, blue line indicates prey, green line indicates predators. B. Continuous generation system. Organisms with continuously occurring births and deaths have a overlapping generations such as several prey and vertebrate predators provide good examples. The continuous generation system used by the predator and prey interaction is described in an independently derived set of mathematical equations given by the Lotka-Volterra 1926. Lotka-Volterra predation equation. In 1925, a physical scientist and mathematician A. J. Lotka, in his work Elements of Physical Biology, suggested the early and very first model of predator-prey interaction system. Independently, A. Volterra developed a similar model of predator-prey interaction in 1926. The model assumed that responses of predator population and prey population are proportional to the product of predator and prey population size. The predator-prey equations given by the Lotka-Volterra describe the interaction between prey and predation as a dynamic biological system. The predator and prey populations follow a non-linear first-order differential equation and represented in pair of equation as dx upon dt is equal to ax minus beta xy, dy upon dt is equal to delta xy minus gamma y, where x and y is the number of prey and predator population and dx upon dt or dy upon dt are the growth rates of prey and predator population in time t. The symbols alpha, beta, gamma and delta denotes the real and positive parameters related to the interaction between prey and predator species. Lotka-Volterra predation equation is based on these assumptions. Predator can eat limitless. Supply of food resource, that is prey, depends on the prey population size. The rate of change of population directly depends on its size. Environment is constant. Inconsequential genetic adaptations for both species. All-time unlimited food supply for prey. The equation is continuous and deterministic, indicate continuous overlapping of prey and predator population. The rate of growth for predator population is given by equation dy upon dt is equal to delta xy minus gamma y. The change in predator population equals to food supply mediated growth minus death of predators. The growth of predator population is represented as delta xy. Delta is different form of beta as the rate of predator population growth is very much different from the rate of predation on prey. Gamma y is the decay rate of predator either due to immigration or mortality. In the prey absence, the predator follow an exponential decay. The solution to equation is periodic and yields a simple harmonic motion where prey population is traced by predation population by 90° in cycle. We can see in figure number 3. The graphical presentation of Lotka-Volterra model is given in figure number 3. This figure represents graphical presentation of predator-prey interaction in model given by Lotka-Volterra. A. Harmonic motion of prey-predator system and B. The combined zero isoclines of model by Lotka-Volterra. The prey isoclines or zero growth curves are horizontal and the predator isoclines is vertical. Both isoclines intersect at right angle. Later, 1935, Nicholson and Bailey recognized the lacunas in the model of predator-prey interaction proposed by Lotka-Volterra and developed a model for a host-parasitoid interaction. The Nicholson and Bailey model involves paired equation as follows, where H and P represent the host and parasitoid population respectively. Lambda is the finite rate in host population increase. A is the rate of parasitism per predator or parasitoid and T is the time. This figure represents graphical representation of hypothetical prey isoclines given by Rosenwing and MacArthur and Lotka-Volterra model. The prey curve is convex in Rosenwing and MacArthur model whereas linear per sec Lotka-Volterra model. The early models given by Lotka-Volterra provide insignificant biological realism and proved unrealistic and thus replaced by other models. Berryman 1992. In 1963, Rosenwing and MacArthur developed graphic models which were regarded as best journal model. In contrast to the unrealistic horizontal prey isoclines of model given by Lotka, Volterra, Rosenwing and MacArthur provided more realistic hump-shaped prey isoclines. Isocline is a fixed boundary at which the rate of increase in the population of prey is zero. It's a boundary between two phases, a zone of prey decrease and zone of prey increase. The prey population size lies somewhere on this prey isoclines when established at the equilibrium. Lotka Volterra assumed the prey isoclines as a horizontal line, but Rosenwing and MacArthur suggested more realistic hump-shaped prey isolines. We can see in figure number 4, hump in the isoclines is caused by the limitation in the increase in prey population. The prey population is constrained by the ecological factors such as diseases, food, shortage, nor social interactions that limit their own rate of increase in population size. On the right side of the hump, the dominant limitation is the intraspecies competition between prey populations that causes the isoclines curve to fall off, whereas on the left side of the hump, predator limits the increase in prey population. The upper limit of prey abundance and the exact shape of the isocline curve are determined by the carrying capacity of the environment and the demographic characteristic of prey. Likewise, prey predator numbers are also limited by certain ecological factors at high predator densities. When the prey densities is low, the predator population is food limited as it depends on the population size of prey. But when the prey population is high, the predator population is limited by their own rate of increase, such as territorial behavior, etc., resulting into predator isoclines. We can see in figure number 5. This figure represents a hypothetical vertical predator isoclines of Lotka-Volterra model. This figure represents Rosenwing and MacArthur model with predator isocline. Color line zone depicts the predator increase in abundance. We can see in figure number 6. Vertical isoclines of predator assumed by Lotka-Volterra suggested that predator population increases only at single prey density. At right line band predator isocline given by Rosenwing and MacArthur. The isoclines is inclined towards right because as the population size of predator increase, intraspecies competition for breeding site and other resources. The shape of predator zero isocline is not same for all predators. Moreover, in case of more efficient predator, the predator is isocline shift to left. The graphic model developed by Rosenwing and MacArthur for the predator-prey interaction is given by superimposing the two isoclines, prey isoclines and predator isoclines. Thus, the effects of single changes to the system of predator-prey interactions can be explored by using graphic model of Rosenwing and MacArthur model. The model of Rosenwing and MacArthur ignored the ratio of prey to predator numbers. To take into account this ratio of prey number to predator number, RDT and Berryman hypothesized a ratio-dependent model RDT 1991. This model proposed that the rate of predation is dependent upon the ratio of predators to prey. As the prey abundance increases, the predator productively increases and conversely, as the prey abundance decreases, the predator productivity decreases. Thus, predator and prey equilibrium both rises with increases in the prey abundance whereas fall with decreases in prey abundance. Predation-Prey System. The behavior predictions of predator-prey system given by Lotka-Volterra model stimulate experimental investigations. Laboratory studies in 1934 Gauss, the Russian biologist, under the constant environmental conditions, reared together a predator and prey, ciliates, Didinum, nostrum and Paramecium quadratum, respectively. He performed two different types of experiments in a simple environment. In one experiment, he introduced predator in a test tube containing clear nutritive medium supporting a population of Paramecium prey. Paramecium was unable to escape from the predatory ciliates Didinium. Prey scrambled to predation. On predation of complete prey population, the predator Didinium died due to starvation. We can see in figure number 7. Thus, from this experiment, he discovered that the predator always experimented the prey irrespective of prey and predator population size. Gauss discovered that the predator population can be maintained only by periodically adding immigrants or prey to the medium. This figure represents prey-predator system between Paramecium and Didinium in oat medium with sediments. Blue line represents Paramecium quadratum, pink line represents Didinium nostatum. In another experiment, Gauss introduced predator into the test tube containing sediment at the bottom, acting as a site for prey to refuse from the predator. Following consumption of all the available prey and predators died from the starvation. Meanwhile, prey that refuse in the sediments increases in number and eventually spread over the medium. By periodically introducing predator to the medium, Gauss could be able to maintain both the population we can see in the figure number 8. This figure represents prey-predator system between Paramecium and Didinium in oat medium without sediments with immigrants. Blue line represents Paramecium quadratum, pink line represents Didinium nostatum. Gauss experiments did not support Lotka-Volterra predation model and their predictions. Gauss concluded in his model that predator-prey interaction cause overexploitation of prey population followed by decline in predator population, and the predator-prey system could not be maintained only by periodically introducing the immigrants. We can see in figure number 9. Inspired from the work of Gauss, C. Huffaker 1958 established a complex laboratory environment to eliminate the possibility of self-extermination of predator-prey system. He introduced a mite, Typhlodromus occidentalis, as predator and a six-spotted mite, Eotetranychus sexmaculathus, as prey which feeds on oranges. Both predator and prey reproduce parthenogenetically. On trays, he established experimental population containing 4 rows with total 40 cells, 10 cells each row. He placed rubber ball in some cells where oranges of same size of remaining cells and thus regulate the pattern of food dispersion, total food availability to control the distribution and size of prey population. From his experiments, he concluded that For prolonged time, the low population size of prey leads to collapse in predator population. The immigration of prey is essential to eliminate the occurrence of self-extermination in the predator-prey system. The intensity of predation has relatively low influence on the period of oscillation is respect to predators searching for prey and prey dispersal complexity relationship along with the time required by prey to recover and repopulate followed by predation. This figure represents predator-prey system between Typhlodromus occidentalis predator and P. tetranychus sexmaculathus prey. Field studies. The field studies are supported by the laboratory experiments around 1839 and ornamental species of cactus called as prickly pear cactus was introduced into Australia from America. These species of cactus, like other invasion species, escaped from cultivation and rapidly spread to nearly 60 million acres in New South Wales and Queensland. The control, the prickly pear, a cactus feeding moth, was liberated from the South America. The moth multiplied, spread, and removed the prickly pear plant until the cacti existed only in widely distributed small colonies we can see in figure number 10. The decline in the plant also causes decline in moth. As the plant population increases, so did the moth until the plant colony declines again. However, few scattered seeds got established in new areas and formed new colonies to maintain the existence of species and predator-prey system. An unsteady equilibrium is maintained by the moth-cactus relationship wherein increase in the abundance and distribution of cactus plant subsequently trigger increase in number of moths and causes decline in cactus. These complex from laboratory experiment and field studies illustrate predator-prey system. This figure represents Cactoblastis cactotortum moth on cactus and its larvae feeding on cactus. Functional and numerical response. In 1959, Holling suggested that as the prey density increases, it leads to increase in predation traits due to two effects. Functional response, where the consumption rate of predator increases in presence of high prey population density. Numerical response, in which increases in prey density leads to increase in predator population density. Functional response. There are three types of functional response curve which relate density of prey population to prey consumption by single predator per unit time. Type first response curve. It's a rare form of response in nature. For example, in filter feeders, it's an essential exponential relationship between prey and predator, and this increase in prey consumption by predator continues till reaches saturation point where predator cannot eat maximum from the that rate. Type 2 response curve. Type 3 response is also common in nature, depicting logistic increase in the rate of consumption on increase in prey density that level off to a plateau. This type of response destabilizes the prey population. We can see in figure number 11, type third response curve. It's a common type of response in nature, for example, rodents and whales. At low population density, the rate of consumption increases at decelerating rate increases at slow rate. followed by sigmoid curve pattern of increases till it reaches asymptote satiations. The rate of consumption is dependent on two factors: A. The searching or locating a prey, and B. Handling time of prey, capture, killing, and eat. At low population density, the searching for prey is important and predator kill prey at constant effort fashion, while at high density of prey searching for prey becomes easy but handling time is limiting factor, and thus the rate of consumption increases slowly. Subsequently searching is not required and rate of consumption levels off at the maximum rate. It's a stabilized response attributing to the density dependent amount of prey consumed. We can see in figure number 11. This figure represents three types of functional response curves. Numerical response means increase in predator density on increase in prey population density due to the direct responses, that is A. On increase in prey density, the fecundity rate of predator increases, and B. Aggregation response, another type of numerical response, may occur due to immigration of predator in response to the increase in prey density. As the prey population size increases, the consumption rate of predator increases, leads to high fecundity rate and low mortality rate. B. Aggregation response. Predators aggregation in prey hotspots is called aggregation response. This type of response increases prey predator system stability. In 1966, Hazel proposed three basic types of numerical responses: A. Positive or direct response, in which predators population increase in response to increase in prey density. B. Negative or reverse response, in which the number of predators per unit area declines in response to increase in prey density. And C. No response, in which the predator population, unaffected by the increase in prey density, remains proportionally same. Foraging theory. In nature, different biological factors such as intraspecific or interspecific interactions and physical factors such as landscape, structure, etc. causes heterogeneous distribution of food. Therefore, in ecology, the fundamental objectives persisted for long to study and understand that how predators exploit food patches and effectively search for prey in the patches. Keeping in mind this objective, sequence of optimal foraging theories was developed by several ecologists. The optimal foraging theories provide basis for the interpreting complex predator-prey interaction via testable hypotheses. The assumption made by the optimal foraging theories was that predation activity is optimized to maximize the rate of net energy gain. Among various biological theories on animal behavior, the most extensively developed optimization theory is optimal foraging theory (OFT). A patchy area is an area with high resources compared to average environment. Assume that the prey on which the predator feeds upon is distributed in the patchy area, then the predator spends its foraging time in searching and traveling between patches and rest in handling prey within these patches. Furthermore, while the predator searches for the prey, predator depletes the number of prey within the patches and gradually With time, the benefit of staying in that patch declines. According to optimal foraging theory, the predator may face four type of difficulties. A. The choice for optimal diet. B. The choice for optimal patches. What type of patch should be searched? C. Optimal departure time from the patch, and D. Choice of optimal movement between patches. In order to maximize the foraging efficiency of predator, the predator should make decisions assumed by the optimality approach as per optimal decision. So student, let us summarize what we learned in this module. When one organism consumes other living organism, the interaction is called as predation. It's a type of positive-negative relationship where one individual is benefited at the expense of other. The broadest manner of predation includes parasitism and herbivory. The predator-prey interaction of organisms with discrete and continuous generation systems have been described by the mathematical models. The continuous generation model for predator-prey interaction is given by many ecologists. The very first but unrealistic model is given by Lotka Volterra, followed by more realistic model given by Nicholson, Bailey, Rosenwing, MacArthur and RDT Berryman model. All these mathematical models in ecology predicted and concluded the oscillations of prey and predator population. The resulting oscillation from the interaction may be unstable, damped, or stable. The predator-prey relationship causes two types of responses, namely functional response and numerical response. Predator may consume more number of prey on increase in density of prey population called as functional response. Predator may increase in number in response to increase in prey population called as numerical response. The functional response can be of three types: type first, type second, type third response. In type first functional response, the number of prey consumed per predator is directly proportional to the increase in prey density until it reaches maximum. In second type functional response, as the prey density increases, the number of prey consumed by the predator increases at the deaccelerating rate until reaches maximum. Type third functional responses, the predation rate increases slowly as the prey density increases, later following a sigmoid curve until reaches a asymptotate. Numerical response includes aggregative response in which as the prey density increases in its attracts the influx of more number of predators to that area. Furthermore, numerical response also refers to increase in the rate of growth and development, natality and mortality rate alters of predator population. The food is distributed non-uniformly in the habitat, resulting into the patches where prey occur. Thus, in terms of profit relative to the net energy gain, the predators spend time in hunting prey occurring in patches with more profit rather than the patches with the abundance of prey. This behavioral study of predators leads to development of concept of foraging theory. The optimal foraging theory OFT is a strategy adopted by the predator to consume its prey in such a manner that it should gain a maximum rate of energy. The optimal foraging theory includes the choice of optimal diet by the predator, the choice for optimal patch, what type of fish should be searched, C. Optimal departure time from the patch, and D. Choice of optimal movement between patches. Thank you.